Hi everyone, welcome to your first online lecture. I hope you're enjoying the snow and staying safe. Today we're going to be talking about chapter 16, which, as I said in lecture last time, is the first chapter in unit number 2. So again, this chapter will not be on the exam next Thursday. We're going to start today by talking about what a gene is and what its function is. Then we're going to be moving on and discussing something really essential to this course, and especially unit number 2, which is the central dogma of molecular biology. Then we're going to move on and talk about the genetic code. And then finally, we're going to readdress the notion or concept of mutation, okay, at both the single nucleotide level and also at the chromosomal level. So let's just briefly summarize what we know about DNA's structure before digging in to what a gene actually does. The primary structure of DNA is the sequence of deoxyribonucleotides, and we know now that those are the 4 nitrogenous bases, and they are A, adenine, T, thymine, C, cytosine, and G, guanine. Okay, and we also said in a previous lecture that it is the sequence of those nucleotides in which the information storage capacity of DNA lies. We then went on to talk in detail about the secondary structure of DNA. Watson and Crick noted that the 2 strands of DNA are oriented anti-parallel to each other. So here we see that we have the 5' end on one side on this left-hand strand going from 5 to 3, and then the opposing strand to which it is hydrogen bonded runs from 3 to 5. We also explained that the bonding between these 2 strands, the glue that holds them together, are hydrogen bonds. So between A and T, we have 2 hydrogen bonds, and between G and C, we have 3. It is always that way. And remember that Watson and Crick's discovery, quote unquote, really was only possible in light of the evidence from Edward Chargaff. Remember Chargaff's empirical rules that the number of pyrimidines and purines had to be equal, and then Rosalind Franklin's discovery that DNA's shape is helical. So Watson and Crick took that information and then built on it and discovered that DNA is anti-parallel and that the bonding is complementary from purine to pyrimidine. So we all have an intuitive idea of what genes do. We say that this and this characteristic is encoded by genetic information. But we're going in this next unit to say in a more precise way what it is that DNA actually stores. Okay? So I said on the previous slide that the information lies in the specific or unique sequence of nitrogenous bases. But what kind of information does DNA have? Well, we now know that the true definition or true technical definition of what a gene is that it is a segment of DNA whose sequence specifies the instructions for one gene product, which is either an RNA molecule or a protein. Okay, so a gene simply provides the instructions for either one RNA molecule or, more frequently, DNA encodes for one messenger RNA, which then codes for one and only one protein molecule. But this link between DNA, RNA, and protein wasn't so obvious early on in the history of molecular biology. So there was one key experiment in particular by these 2 scientists, Beadle and Tatum. They designed an experiment that really ingeniously discovered that a DNA molecule codes for one and only one protein product. Like a lot of studies in genetics and molecular biology, they were able to understand the function of genes by inducing mutations into these genes and seeing what the resulting effects would be. specifically in the phenotype, which we haven't defined phenotype, I think, yet in this class, but the phenotype is the visible characteristic that is expressed by or is derived from the genotype, which is the genetic sequence. So here are the set of genes that these two scientists studied. They studied a pathway in the formation of the amino acid arginine. And if you remember from class on Monday, arginine was one of the basic amino acids. And remember that the basic amino acids at most pHs tend to have a positive charge because of that extra proton molecule. So arginine is a charged amino acid. So what they ended up doing was they introduced a mutation into each of these three genes that they thought coded for these three enzymes. So if you look at the next slide, here is a more detailed depiction or summary of what they did. Like a lot of the research boxes that you see in your texts, this is something that you're going to have to probably take a closer look on your own. But I'm going to go over just the main points here so you get a general idea of what they proved. So their hypothesis was that each gene, so each sequence of DNA, contains the information required to make one enzyme, and only one enzyme. So this is known as the one gene, one enzyme hypothesis. The alternative or null hypothesis was that genes do not have this one-to-one correspondence with enzymes, that a gene can code for more than one enzyme. So how did they go about testing this hypothesis? Well, what they ended up doing was growing bacteria of this particular species in 4 different growth media. And they mutated 3 different genes, which we're going to be calling enzyme 1, enzyme 2, and enzyme 3. Now, how this biosynthetic process works is that we have some sort of precursor molecule, and enzyme 1 takes that precursor molecule, alters it to get this next molecule, precursor molecule, ornithine, another enzyme acts on that, converting it into citrulline, and then there's a third and final enzyme which converts citrulline into arginine. So there are 3 precursor molecules that lead up to arginine, and the only way that one precursor molecule is converted into another is with the help of these enzymes. And remember that enzymes are cellular catalysts, right? They lower the activation energy, allowing reactions to proceed much more quickly. So in the first growth media, so this one in kind of this light kind of tan color, there were no precursors supplemented in that solution. So there was no ornithine, no citrulline, and no arginine. So they looked at what happened in each of the 3 mutated cases. So with here, there's a mutation in enzyme 1, 2, and 3. And the results here make obvious sense because if there is no precursor in solution and no ornithine, enzyme 1 can't convert the precursor into ornithine, right? The precursor molecule here is the substrate, and in order for an enzyme to achieve its function or to execute its function, it needs to act on a precursor. So if we have a solution which is not supplemented with that precursor, we're not going to go from the precursor to ornithine to citrulline to arginine. So this mechanism or this pathway never gets underway. So that was true for all 3 gene mutations, okay? But something different happened in the second supplement tube, which contained ornithine only, okay? We see that with the mutation in enzyme 1, which is this ARG1, we do see some growth, okay? Which means that arginine was synthesized. So wherever you see growth here, that indicates that arginine is synthesized, okay? Because remember, arginine is an important amino acid for the development of proteins. So if arginine is lacking in a cell, it is unlikely it will proliferate or reproduce. So look here that there was growth with the arginine 1 mutation when ornithine was present, but not growth for the other 2 mutant types. What the 2 scientists concluded was that if growth or arginine production worked here, that must have meant that these two enzymes were functional. Since we had ornithine present, the enzyme that gets us from ornithine to citrulline, that was working. And the enzyme that gets us for citrulline to arginine must have also been working, otherwise we wouldn't have had any arginine production. So that means by process of elimination, if the mutation is not in enzyme 2 or 3, then it has to be in enzyme 1, and therefore the function of enzyme 1 is to go from this precursor molecule to ornithine. Okay? So that evidence here in this first row, that supported the one gene, one enzyme hypothesis, because this particular mutation had an effect on only one enzyme, enzyme number 1, and not enzyme number 2 and 3. If you look at the next row, you see that arginine 2 did not have growth when ornithine is present, but it did have growth when citrulline is present. Now, if arginine was produced when citrulline was present, that must mean that enzyme number 3 is working, because the only way to get to arginine is if citrulline is converted by enzyme 3 into arginine, right? So we have growth there, but we didn't have growth with ornithine because that would indicate that there's a mutation in enzyme number 2. Okay? So if enzyme number 2 is not working, then even if ornithine is present, we're not going to move or go from ornithine to citrulline. But if the next precursor is present, the one downstream, one molecule over citrulline, when citrulline is present, then we have an active enzyme 3 and arginine is produced. With the third and final case, we see that there's no growth in either ornithine or citrulline, okay? And that means that if there's no growth in those two, then the step from citrulline to arginine is not happening. So that would imply that enzyme 3 is the enzyme that is mutated when there's a mutation in the ARG3 gene. Okay? So all in all, the takeaway, and again, I really recommend you looking at this again, and if you have any confusion about it, please don't hesitate to approach me in office hours or at the end of lecture on Monday. But basically what this experiment really definitively proved was that these three genes, ARG1, 2, and 3, they code for three separate enzymes which have three distinct functions. So, conclusion, the one gene, one enzyme hypothesis is supported. And after this experiment, that one gene, one enzyme hypothesis became established scientific fact. So how does this get us at the central dogma of molecular biology? The central dogma of molecular biology states that information in DNA is transferred to an information storage molecule known as RNA, and then the information in that storage transport information transport molecule is transferred into a protein product. So it is the DNA that is the genotype, the sequence of nucleotides, and the protein, which is the sequence of amino acids. That is what creates the phenotype for all of the traits, or all the physical traits we have. Two terms that are going to be essential for this second unit are transcription and translation. Transcription is the mechanism by which the information in DNA is converted into information in a storage molecule known as messenger RNA, and then the process in which the information in messenger RNA is transferred into a protein product, that's known as translation. The enzyme responsible for transcription is known as RNA polymerase, and that makes intuitive sense because remember that when we were talking about DNA replication in our last class, the enzyme responsible for executing that mechanism was DNA polymerase, right? So if we're making DNA, we're going to use DNA polymerase. If we're making RNA, we're going to be using RNA polymerase. The enzyme responsible for synthesizing proteins, that's referred to as a ribosome, and we'll learn more about ribosome structure in this unit, but a ribosome is part ribosomal RNA, so part RNA, and it's also part protein. The technical term for that is ribozyme. But we'll get into more of that when we have our chapter on transcription and translation. How I remember these two terms, sometimes people mix them up, transcription and translation. Just remember that when you transcribe something, you write it out. So here, picture DNA is, say, one language, English, but if you make a copy of that language on a separate piece of paper, let's say, you're just writing it. It's in the same language, English, but it's just on a different piece of paper, okay? But when you go from RNA to protein, translation, we know that translate means to change language. So if mRNA is written in English, then let's say the protein product would be in Spanish or some sort of other language. To put it in other words, so that was just kind of a pictorial or real-world application illustration, but just remember that DNA consists of nucleotides or deoxyribonucleotides. mRNA also contains nucleotides, but they are not deoxy, they're just ribonucleotides. But still, they're so close in structure that they really are kind of chemically, while there are some important chemical differences between them, they are fairly similar, especially in comparison to proteins, which are much different, have much different chemical properties, as we saw in the lecture on proteins on Monday. So here, I already just said these definitions, but here is another way of stating transcription: the process by which hereditary information in DNA is copied to RNA, and then translation, the process in which the order of the nucleotide bases is converted to the order of amino acids. Here's what it looks like in the cells. So one thing you need to remember about eukaryotic cells, and remember eukaryotes include all animals, all plants, all fungi. Eukaryotes have a membrane-bound nucleus, right? And DNA, as we all know, is housed within the nucleus. DNA never leaves the nucleus and enters the cytoplasm. Protein synthesis, however, occurs in the cytosol or cytoplasm. So you might ask, how is it that the information in DNA is transferred from the nucleus into the cytoplasm? And the answer to that is simple. That's where messenger RNA comes in, and that's where that word messenger comes in, or was introduced, because it has a kind of transportation or carrying function. So transcription, DNA to RNA or mRNA, occurs in the nucleus, and then that messenger RNA molecule literally leaves the nucleus, exits it through a nuclear pore, and then it hooks up with a ribosome, and then protein synthesis or translation is executed. Again, here's just another way to summarize the central dogma. Again, the central dogma states that DNA is transcribed into messenger RNA, which is then translated into protein. DNA is our genotype, the hereditary information, and the phenotype is the result of the protein product, and that is the expressed physical trait or characteristic. Remember that genes can come in different forms. So different versions of the same gene are referred to as alleles. So alleles are variants of a gene, and they have slight differences in DNA sequence. So they code for this same trait, but the protein products, since the DNA is different, the protein products are going to be slightly different, and the physical phenotype or expressed trait is also going to be different. So on this next slide, we have a nice example where we compare two mouses. So we have a forest mouse here, dark-skinned on the left, and then we have a beach mouse, light-skinned on the right. They are similar in almost all respects. The only difference is the color coding of their fur. What leads to that difference? It turns out that the cause of that difference is a change in one DNA nucleotide. So if you look at this stretch of DNA here, we see that it is almost identical with this stretch of DNA, except for this one base. So whereas here we have a guanine G in the first position, here we have a adenine in the first position. And what ultimately happens is, if the DNA is different, that leads to a change in the RNA. And as we can see in the protein product, in the original sequence, we start with an arginine, which we know is positively charged, basic, and that's replaced with cysteine, which is neutral but polar. And since they have different chemical properties, that's what ultimately leads to the different physical expression or different phenotypes. So at the end of this slide presentation, I'm going to talk about mutations, but this particular mutation, without getting ahead of ourselves, it is only different at one point and hence is called a point mutation. And something that slight can have that big of an effect on expressed phenotype. Now, we call it the central dogma, and the word dogma kind of has these almost a religious overtone. Actually, that's where the kind of the word comes from, church dogma, right? So dogma is like a kind of a serious law, something that can't be broken. But it turns out that the central dogma is not absolute. So with time, as molecular biology has matured over the years, scientists have found some exceptions. So the basic scheme for central dogma is that, again, information stored in DNA is transcribed into an information messenger called RNA, messenger RNA, and then that's translated into a protein product. So DNA to RNA to protein. But occasionally, the process stops at RNA. So not all genes code for proteins or functional proteins. There are some RNA molecules that are functional. So the process is just transcription from DNA to RNA. And the two main types of RNA that have functional significance in cells are transfer RNA and ribosomal RNA. And those are tRNA and rRNA, respectively. These are both involved in the process of translation, which is protein synthesis. Protein synthesis would not be possible without these two RNAs. And again, we'll be getting into more detail about these when we talk about transcription and translation, which is the first chapter we're going to review or go over after the exam on Monday. So that's one exception to the central dogma, that sometimes the process doesn't go all the way to the protein level. It just stops at RNA, so only transcription. With some organisms, there is a process known as reverse transcription. So forward transcription is DNA to RNA, but reverse transcription is RNA back to DNA. And there are many retroviruses, so that's a class or group of viruses. They actually use this process, RNA to DNA. Okay, so it seems unusual, but among viruses, it's a mechanism that is common. Shortly after the experiment that I reviewed just a handful of slides ago, once the one gene, one enzyme hypothesis was really established, then scientists, specifically molecular biologists, wanted to understand how much of the DNA codes for one amino acid. Because we know that DNA is simply a sequence of deoxyribonucleotides. When it's transcribed, we have a sequence of ribonucleotides, and then from the ribonucleotides, we get a sequence of amino acids. But what is the correspondence between nucleotides and amino acids? How many bases, nitrogenous bases, code for one amino acid? Does one nitrogenous base code for one amino acid? Is it two for each amino acid, 3, 4, etc., etc.? So how does this process actually work? Well, one scientist who really was instrumental in deciphering the genetic code was George Gamow. Okay, now he predicted that one amino acid is encoded by 3 nitrogenous bases. The question we want to ask is, how did he know, or how did he decipher that was actually the correspondence, 3 bases per amino acid? Well, he basically performed a kind of thought experiment, mathematical thought experiment like this. So let's say that there actually was a one-to-one correspondence between nitrogenous bases and amino acids. So for every base you have in DNA, you have one amino acid. Well, we immediately run into a problem because we learned on Monday that there are 20 unique amino acids, but there are only 4 nitrogenous bases that make up DNA. So the one-to-one correspondence couldn't work just from a practical standpoint because 4 is so much less than 20. There's just not enough nitrogenous bases. Now, if there were 20 nitrogenous bases in DNA and 20 amino acids, then it would make perfect sense. But that's just not the reality of DNA. We just have 4 bases. So what about if we had a 2-to-1, so 2 nitrogenous bases per amino acid? Well, we run into the same problem. The situation is a little bit better, but still not perfect. Because if we have 2, then we have 16 possible pairings. So that would code for, say, 16 unique amino acids, but 16 is not close enough to 20. So it's still not enough. So then it seemed logical that if we had 3, then we would have 64 possibilities, right? 64 combinations of 3 nitrogenous bases, and that's more than enough. So the problem seems to be solved, but still we don't have, we have 64, and 64 is quite a bit more than 20. So what's going on with the additional 44 triplets? Okay, doesn't it seem a little bit kind of overkill? Don't we have too many combinations? Well, let's see how that dilemma was resolved. So again, George Gamow, he predicted that the genetic code is based off of triplets, okay? And we have 64 different possibilities for 20 amino acids. Here is the actual genetic code, and this is what it looked like when it was first deciphered, and it really hasn't changed. So on the kind of left-hand axis, we have the possibilities, the 4 possibilities for the first base in the codon. On the top, we have the 4 possibilities for the second base, and then on the right-hand side, we have the possibilities for the third base. So we have 64 combinations here. And the first thing that is obvious when we look at this table is that there is more than one triplet, and the word again, instead of triplet, we're going to use the word codon, that's the technical term. But we have more than one possible codon for an individual amino acid. So look here at serine, we see that there are 4 different ways to create a serine molecule. Not only that, but actually if you look over here, there are 2 more. So there's actually 6 possible ways to synthesize a serine amino acid. So the term for that, the term for the repetitiousness, if that's a word, or the repetitiveness, sorry, that's a real word. The repetitiveness of the genetic code is referred to as redundancy. So we say that the genetic code is redundant and that there are multiple, not for all of the amino acids, but for many of them, there are multiple codons that code for that particular amino acid. Okay, so that word redundant is on this next slide. All amino acids except 2 are encoded by more than one codon. The next important property of the genetic code is that it is unambiguous, which means that one codon never codes for more than one. So going back to our serine example, here we have the codon UCU. UCU will only code for serine, so it does not code for any other amino acid. I'll skip this one for a brief second, but the other one I really want to emphasize is that the genetic code is conservative. What is meant by that? Well, notice that for serine here, that all 4 of these codons start with UC. So UCU, UCC, UCA, etc. Okay? So what we mean by conservative is that for most of the amino acids, if the first 2 letters are the same, then regardless of the third letter, those codons are going to code for the same amino acids. So if you start with UC, all of the one actually, in fact, all of the amino acids that start with UC, they all code for serine regardless of this third base. Similarly, if we look down to proline, we see that all 4 of these amino acids start with CC. Regardless of what happens in position number 3, all of these, if you start with CC, you're going to code for a proline. Okay, so this is what we mean by conservative. And the fact that this, I don't think I have this mentioned in these slides, but it will be mentioned later in transcription translation. But there's a principle known as, there's something called the wobble hypothesis, which states that that there's a certain flexibility in the third base, meaning that as long as the first 2 are the same, it doesn't even really matter what's in the third position. I'll mention that again. It just comes to mind now when I look at this, but I'll mention the wobble hypothesis again when we talk about translation in just a couple of lectures. The last one of these properties I didn't mention was universal. So in general, the codons we see in this table go across species, go across the tree of life. But there are some exceptions, but by and large, the code is universal. So for instance, the amino acid that always starts proteins, AUG, which codes for methionine, that holds true for essentially all organisms we know. But again, there are exceptions. The next thing that's important to talk about with the genetic code is this notion of reading frame. Okay, so what do we mean by a reading frame? Well, take a look at the bottom of this slide. Here I've written, I have a jumbled up sentence, and then I space it out in triplets in 2 different ways. Okay, so here we have the squished phrase, but if I start here and count triplets, so the, and then start my next triplet 3 bases over, the fat cat, and keep doing it, the sentence means the fat cat ate the rat. So you can think of this smushed up sentence as the genotype, and then here we have the phenotype, which is the coherent protein product, the fat cat ate the rat. But the reason why this sentence is coherent is based off of the reading frame I established at the beginning. Because I said that T is the first letter in the codon, I say that this represents 1 codon, so the next consecutive codon, the next triplet, would have to be FAT, fat, which is a coherent word. But we could have started with a different reading frame. What if I didn't start on this T, but I started on H? So my first 3 letters would be HEF, hef, which is a nonsense word. But to be consistent, if I establish the reading frame here, then the next 3 consecutive letters would be ATC, the next would be ATA, and so forth. If I establish the reading frame based on this letter, I would end up with a nonsense expression. And at the very end of this lecture, I'm going to talk about a specific mutation that leads to these kind of nonsense sequences, and it has to do with a shift in reading frame. So we'll get to that in just a handful of slides. But the bottom line I want to get across on this slide is that reading frame really affects the final message, the final protein product. And how many possible reading frames are in a single strand of DNA? Well, generally for eukaryotes, there's 3. Because if you think about it, we could have started on T. If we start on T, we get our coherent the fat cat. If we start on H, we get this incoherent expression. And if we started on the 3rd letter, we would likewise get an incoherent expression. So the only one that makes sense is if we start right here. And going back to what I said about AUG methionine being the first amino acid, that's how the reading frame is established. So the ribosome kind of locks on to AUG, and then it reads in triplets after AUG. So AUG is significant because once it's recognized by the protein synthesis machinery, it establishes the proper reading frame. Again, this lecture is just an introduction to central dogma and transcription and translation, but we're really going to get into detail in the next couple of lectures after the exam. So again, how are we able to know which of the 3 reading frames is correct? Well, these 2 scientists, Nierenberg and Leader, they deciphered the genetic code, and they were able to figure out that every protein started with that AUG. So that indicated that that's where the reading frame is established. And they also noticed that the end of a protein sequence always ends with these three codons, UGA, UAA, and UAG. Whenever these pop up in a sequence of messenger RNA, that signals, or they signal, the termination of that protein. So these are called stop codons. This slide, I don't want to get into too much detail here, but one of the things that you'll have to do on your second exam is you'll be given a sequence of DNA like this. And from that sequence, you'll have to predict the messenger RNA and predict the protein product. Here on the right, they say that you can actually go backwards, but basically what you're going to see on the test, and we'll go over an example, especially in discussion with your discussion leader, you'll go over this. But based off of this DNA, you'll learn how to predict what RNA will look like, and then you'll predict what the protein product will be. So you'll actually be transcribing and translating your own genes. That will certainly come up in a homework assignment, homework number 2, and you'll also see it on an exam. It's something that really is important. It's an important concept that we want to get across in this class. Okay, so now we've reached our last topic for today. So what have we done thus far? I started off by talking about what is a gene and how the function of a gene was determined, right? We looked at that experiment which determined that there is a one-to-one correspondence between gene and protein, the so-called one gene, one enzyme hypothesis. Then we talked about the central dogma of molecular biology, DNA to RNA to protein, with occasional exceptions. Then we just finished talking about the genetic code and its essential principles. And now we're going to return to the topic of mutation. We talked about mutation in our unit on our chapter on evolution. I defined mutation, and your textbook says it in different words, as mutation is the driving force of evolutionary change. And a mutation is any permanent change in an organism's DNA. So by definition, mutation means change in DNA. Okay. So if you change the DNA, you change the genotype of that organism. And whenever you change the genotype, you're going to be creating new alleles or new versions of those genes. And remember, from a natural selection standpoint, if we create new alleles, some alleles, or particular alleles that lead to some sort of survival or reproductive advantage, they will be propagated more frequently, leading to more offspring with those alleles, and hence a change in allele frequency, and thus evolution. Now that, as a class, we know a lot more about the structure of DNA, we can be more precise about how we define mutation. So the first type of mutation I want to talk about is point mutation. Point mutations typically arise when an error is made in DNA replication. We saw a slide like this in our lecture on DNA replication. Here we have our original DNA strand. It separates, remember helicase unzips it, but when the replication takes place, we see that an error is introduced. Normally, either the proofreading mechanism of DNA polymerase, it will remove this incorrect base, or we also learned about mismatch repair enzymes, which also remove incorrect bases. But if this mistake is not recognized by either of those two mechanisms, then the mistake is permanent. And if the mistake is permanent, the next time DNA replication occurs, then we get a changed base pair. So the original base pair here was C with G, but somehow a T was mistakenly placed in the DNA sequence. So once the T is placed, then we have A bind to it. So we have a totally new base pairing. The consequence of that mistake, it's also referred to as a base pair substitution, can really be significant. So let's take a look at this example. What if we change the third letter in this codon? So originally we have TAT, which codes for tyrosine, right? Tyrosine is one of the polar amino acids, polar uncharged. We have TAT, but what if there was an error in DNA replication that led to a change, and now the codon read TAC? While the DNA sequence is different, the protein is the same. Why? Well, remember I mentioned that the DNA code, the genetic code, is conservative. Since the first two letters are the same, TA, it turns out that that change in the third position doesn't end up changing the protein. Since the change in DNA causes no change in the protein, we refer to that mutation as a silent mutation. So it goes essentially undetected. It's a change that goes undetected and has no consequence. Okay. Here, in the next example, we do also get a change in the third position, but it turns out that that change actually leads to a change in amino acid. So here we have a codon that the resulting codon leads to a different amino acid. So instead of having tryptophan, we now have cysteine. So tryptophan and cysteine have different chemical properties. So that protein is likely going to fold differently and probably will not function properly. So we call that a missense mutation. And how I remember missense mutation is I refer to it as a mistake. So there is one mistake in this protein, and that one amino acid is different. But note that not all of the amino acids are different, just that one. So sometimes missense mutations aren't much of a problem, but sometimes they are. As with the sickle cell anemia example we looked at, where that was a missense mutation, right? And that led to a deleterious impact. Sometimes these point mutations can lead to the introduction of an immature stop codon. So if the third nucleotide here is changed to an A, then in the RNA, in the complementary sequence, we get UGA, which is a stop codon. So when that stop codon is introduced, then none of these three amino acids here on the right are encoded. So the amino acid is, the protein product is stopped short, it's truncated. And those are usually quite deleterious because if you have a shortened protein, it's certainly not going to fold. And if it doesn't fold properly, and if it doesn't fold properly, it will not function properly. So kind of in order, silent mutations have a neutral effect. Missense mutations are occasionally, but not always, harmful. Nonsense mutations usually have serious consequences. And then finally, frame shift mutations are also, like nonsense mutations, fairly deleterious or harmful. Whereas in the above three examples, we see that one base is substituted for another, called a base pair substitution. So these three, silent, missense, and nonsense, are base pair substitutions. The last, in a frame shift mutation, what happens is that rather than replacing one of the nucleotides is simply removed. So it's just plucked out. So if you look back to the original sequence, we had TAT, TGG. If you removed this G, right, then you would get a sequence that would say TGC. Although that's not what I'm seeing here. I think this might be a mistake. But anyway, the bottom line here with frame shift is notice that once that nucleotide is removed, we see that all of these amino acids are changed. And that goes back to this example I said here, is that here the reading frame is thrown off So if we start with H, we get this kind of really kind of nonsensical phrase. And that's exactly what happens with frame shift mutations, is that if we remove one of the bases, it throws off the reading frame, and then we get a whole different sequence of amino acids, and an entirely different protein. And if the protein is almost entirely different in amino acid sequence, it's not going to function as it should. So these are point mutations which involve either changing one nucleotide or removing one nucleotide. So silent, missense, and nonsense are base pair substitutions, whereas frame shift involves the removal or addition of a nucleotide. Those are what I call small scale mutations. They're not small in the sense that they're not significant. As I've just described, they can have big effects, but they're small in that they just involve either the addition, change, or removal of a single nucleotide, or sometimes a couple of nucleotides. So a lot of mutations, though, can occur at the chromosomal level. So we're talking about long stretches of DNA, because remember, a chromosome is a large fragment of DNA that contains many, many genes. So a chromosome is a kind of a macromolecule that contains many genes lined up. So these are chromosome level mutations, are large in scale, and generally they fall into two different classes. We have polyploidy, and then we have aneuploidy. Polyploidy is not something that commonly occurs in humans, but it involves an increase in the number of each type of chromosome. We as humans, for example, have 46 chromosomes, and we say that we're diploid because we have 23 unique chromosomes, but since we receive a set from each of our parents, that's 23 times 2, giving a total of 46. We're diploid, but there could be a situation, if you could picture a human whose genotype is triploid, where they had 3 sets of 23. And if I do my math right, I think that would be 89 chromosomes. So that would be substantially more. That would have enormous consequences. I'd be surprised if a fetus with that genotype would even survive. What's more common in humans and often leads to certain disorders is aneuploidy, where there is addition or deletion of a chromosome. And the classic example of this would be Down syndrome, which is known as trisomy 21, where individuals, through an error in meiosis, end up having an extra chromosome 21, hence trisomy standing for 3. So that would be an example of aneuploidy. In addition to polyploidy and aneuploidy, there are 2 other types of chromosome level mutations, whereas polyploidy and aneuploidy involve whole chromosomes. These 2 types of mutation, inversion and translocation, involve just parts or segments of chromosomes. So an inversion occurs when a whole section of a chromosome breaks off of that chromosome and then rotates and reconnects. So as you can imagine, that would kind of totally change the genetic sequence and lead to some damaging consequences. Translocation is not always deleterious, but it can be. And this occurs when a section of a chromosome breaks off from one chromosome and then attaches to a totally different chromosome. A healthy example of this happens during meiosis, and we'll talk about this later in the semester, in fact, the last unit, so after spring break. But during meiosis, there's a process referred to as crossing over, where when the chromosomes line up before splitting into 2 different cells or dividing into 2 different cells, chromosomes cross over and exchange genetic information. That would be a healthy example of translocation, which increases genetic variety. But we can imagine that translocation could also lead to a deleterious effect like inversion and both polyploidy and aneuploidy. How were scientists able to discover these large-scale or chromosome-level mutations? Well, often they were discovered by looking at what is called a karyotype, which is a pictorial representation of the entire genome or all the chromosomes of an organism. So here is a sample karyotype. Now this isn't really realistic, but this shows some of the chromosome mutations that we can observe. So for example, here at chromosome number 3, this individual has 3 of those 3 chromosome number 3s, so that would be trisomy 3, in fact. I'm not exactly sure what kind of disorder that would lead to, but this illustrates that point. Here, and for this pair of chromosome number 4, we see that this light blue was probably the original chromosome, but then we see bits of other chromosomes, so that would indicate perhaps some translocation or inversion. Okay, so that's it for this lecture. I think I skipped over one slide, and that was just here, which this slide simply just says that mutations can have one of 3 effects. They can either be beneficial, so that's what we look at when we're talking about increasing fitness in a natural selection context. Sometimes mutations can have really no effect, just like the silent mutations where there's a change in DNA, but it doesn't change the protein. And then sometimes they can be deleterious, as we saw with the sickle cell anemia example, where a missense mutation leads to the sickling of red blood cells and then ultimately cardiovascular problems. So that's all I have for this lecture. That takes us through chapter 16. In the next class, we're going to be talking about transcription and translation. And remember, on Monday, which I believe is the 13th of February, we're going to be having a review on all of the material for chapter 1. So no new material on Monday. We're going to be reviewing for our first exam, which again will be held on Thursday of next week. All right? So see everyone on Monday.